The next speaker I'm very delighted to introduce you to is Andrei Seluanov. He's a professor of biology, and I think I didn't pronounce your last name correctly, so you might do that on my behalf again. He's a professor of biology and medicine at the University of Rochester, focused on understanding the mechanisms of aging, DNA repair, and cancer. And Andrei has made a particular name for himself due to his studies on cancer-resistant mammals. Welcome, Andrei.
Okay, thank you. Yeah, thanks. First of all, I would like to thank Abri for inviting me for this amazing conference. Again, I think it's a unique blend of basic research, clinical research companies, and just longevity enthusiasts in general. You know, it's amazing. Second, I would like to say that I don't have any companies. I'm just doing the basic research. And my ultimate goal is to identify the mechanisms which allow some mammalian species to live much longer compared to the others. We're working with many mammalian species. Actually, in the lab, we have around 56 mammalian species. But today, I'm going to talk about these little creatures, naked mole-rat. And it's already was some discussion today in the morning session about the naked mole-rat. I mean, some people are already familiar about this species, but I would like just to remind this, the naked mole-rat is the mouse-sized rodent. The body mass around 25 grams, the same like mice, but it lives 10 times longer. I put like 40 years plus, because if you remember morning discussion, we don't know exactly maximum lifespan. They could be longer than this. Hopefully, Steve Horowitz in the end will develop his clock, and we'll have the better data about this, or we'll wait more years until all this animal will die. The other thing, in addition to extreme longevity for naked mole-rat, it's famous for its resistance to cancer, to oncogenic transformation induced by the drugs or spontaneous cancer resistance. And it's also resistant to many age-related diseases, and I have the list of them here, some of the arthritis, neurodegeneration disease, or heart problems. And in general, you know, these animals not only live long life, but they also live the healthy life. That's all of us wants to do it. And in 2013, naked mole-rat ended up on a Nature cover, primarily because of our publication, when we found one of the mechanisms which allow these animals to live longer and stay cancer-free. We found the cells of naked mole-rat producing this hyaluronic acid, or hyaluronan, the long linear polysaccharide, which is presented here. It's present in the tissues. And you'll see in this Petri dish, actually, fibroblasts are growing on the bottom of the plate, and you'll see this gooey substance which is produced by the fibroblasts is hyaluronic acid. But this was published a while ago in 2013. What we found is that in paper that the naked mole-rat produce not only huge amount of hyaluronic acid, but also very long form. And if you look on a gel on the right side, that you'll see naked mole-rat size of the hyaluronic acid is above of 6 megadalton. It means we can't resolve exactly the size of that, but it's very large. And how naked mole-rat achieved that high level of very long form with hyaluronic acid present in the tissues is one by the boost synthesis. It means they have the enzyme called Has2, which I'm going to talk in today's presentation, which produced a lot of the HA. And the second, they have the slow degradation process. And both of them leads to accumulation of this HA in the tissues. And the summary of that paper, which was published in 2013, is just in this, presented in this formula. If you take the naked mole-rat cells, which comes from the cancer-resistant long-lived species, and remove the HA by different methods, you'll get the cancer-prone cells. It means the resistance of the naked mole-rat cells is due to the production of hyaluronic acid, and probably leads to a shorter lifespan in mice. Of course, this is an interesting observation, but it's not really practical. And since Steve taught me math, and he's a really great mathematician, I decided let's reverse this formula and see if by addition of the HA into the mice will lead to the production of the resistant mice, in this case very similar to naked mole-rat, which may live longer life as well. And in order to address this question, actually, we generated transgenic mice which overexpress the naked monad HAS2 gene and a very strong CAG promoter in most of the tissues. Actually, I must tell you that overexpression of HAS2 has some negative effect, but it's primarily during the embryogenesis, because when we first try to generate HAS2 overexpressing mice, actually we failed to do it because this embryo will never form. The HAS2 or HA interfere with overproliferation of embryonic cells. And what we did in this system, it was an inducible system. We can inject tamoxifen to induce a Krellox recombination and leads to activation of the HAS2 gene. And usually we are doing the activation at age of 2 to 5 months. It means that's when the mice is almost fully grown. And we start to see the high expression of HAS2. And most importantly, we start to see the accumulation of the HA in the mouse tissues, and you'll see here the data is a bit hard to see. The red stain is represent the overproduction of the HA in naked monad HAS2 overexpressing mice. I must tell you the level of the HA, which is shown here, is much lower than what we observe in naked monad. Primarily because here we just boost the expression, but we still have the high level of degradation enzymes present in the mice. It means we are talking about suboptimal high level of the HA expression, which is lower than normal in naked monad. And what happened to these mice? Of course, we look in the cancer, and we'll see during spontaneous cancer, it means these mice, they start to have a low rate of the spontaneous cancer. This will look post-mortem in all our mouse aging colonies. And we also try to induce the tumor in these mice by using the TPA-DMBA treatment, the classical skin cancerogenesis, and you start to see also reduction of the formation of the papillomas or tumors in these mice as well. And this paper was just recently accepted to Nature. It will be in press in a few weeks from now, and hopefully you'll be able to see it. And what we found when we grow these mice, transgenic mice, when we get them into the age of 25 months of age, you start to see a big difference between the control. First of all, they look much nicer. If you compare these two mice, you can immediately see which one is control, which one is the HAS2 overexpressing mice. It has a much shinier fur, fuller fur as well. They have the less gray hairs, and they look much better in general. But what about the longevity on these mice? We did the longevity experiments, and you'll see this HAS2 overexpression mice, they live a little bit longer, but significantly longer than the control mice. But most importantly, in addition to a long lifespan, they start to see the healthy phenotype. Because here we use the frailty index, it's a plot which is number D, the panel number D. You will see that actually the frailty index goes up with aging, because we are all getting frail, the mice getting frail as well. And when the mice overexpressing HAS2, we'll see the reduction of the frailty. And the similar phenotype was observed in the rota experiments. They were much more robust, the mice which overexpress HAS2, and they also have the much better the grip strength. And we also use the methylation clock to confirm this life extension, and we will see the reduction of the methylation age in mice overexpressing HAS2. It means, yes, they live longer, and most importantly, they live healthier life. And of course, in order to address the questions how we achieve this longer lifespan and resistant to the all age-related pathologies, we did the RNA-seq analysis of 5 different tissues. And we compared the profile of transcriptomes of young and old mice as well. And what we'll see that the HAS2 or naked monad HAS2 overexpressing mice, the transcriptome is shifted toward the younger transcriptome compared to the same mice as well. And if you compare it, we build also transcriptome analysis based on different mammalian species of long-lived and short-lived, and we have the transcriptome which associates this longer lifespan, the shorter lifespan of different species. We published this this data in Cell Metabolism, and again we will see the shift of naked monad HAS2 overexpressing mice toward the longer life organism, which is actually naked monad as well. That means we are mimicking the same situation in the mice what we observed previously in the naked monad. And of course, toward the mechanism of this, we also looked at an inflammatory response, because inflammation is associated with aging. We are talking about inflammatory aging phenotype. Inflammation comes really associated with aging and triggers many age-associated diseases as well. And you'll see here, it's one of the examples. We look on the liver and the females, but we see it in many different organs as well. That in the control mice, which is CRE-ER, this is control animals, we'll see induction of the inflammation with aging, age of 24 months. And host overexpressing mice, they have a reduced inflammation compared to the same age group of the control animals. And we look at the profiles of different secretory cytokines in the plasma, and also we see reduction upon overexpression of HAS2. It means it's really have reduction of the inflammation. And if you are talking about the association of inflammation and hyaluronic acid, I would like just to remind you that the size of the HA is really important for its function. Because if you are talking about the small molecules, which is like a 10 kilodalton or 100 kilodalton in the size, and then we are talking about the molecules which induce the inflammation. They promote the cancer and induce the metastasis. It means the short form is bad because it gives the inflammation and the cancer. And the long form, like in human, we are talking about 4 to 5 megadalton in size. It leads to the homeostasis and works as anti-inflammatory factors. And super long molecules, what we observe in naked model at 6 to 12 megadalton in the size, we'll see the multiple positive effects. They reduce inflammation, reduce the cancer burden as well in these mice, and probably leads to longevity. It means size is absolutely critical for this. And what we observed in this host overexpressing mice, it's not only the amount of the HA goes up, but also the length gets increased. Usually HA is associated with protection from free radicals, because HA binds the free radicals and protects the cells from this. In order to confirm this, we isolate the cells from HAS2 overexpressing mice, and we expose it to hydrogen peroxide, the free radicals, of course. You'll see that the cells from HAS2 overexpressing mice, of course, is more resistant, which is relatively obvious. But also we try to induce the inflammation mice by using the LPS. That's like we're mimicking in this case, like the bacteria trying to penetrate into the organism. And what we'll see again, the reduced inflammation in the host overexpression mice. Again, that's one of the mechanisms how the HA is working through the reduction of inflammation during the aging process. The other interesting phenomenon what we observed on these mice, when we look at the old host overexpressing mice, we start to see that the gut function gets improved in host2 mice. And how we observe it, because with aging, the mice start to have the leaky gut. It means if you inject it at the fluorescent level, or if you gavage the fluorescent level to the animals, this fluorescent level end up in the blood because it's more leaky. But in young animals, it stays inside of the gut because the gut has a really intact epithelium. And here you'll see on the left panel, actually you'll see that the permeability of the gut epithelium goes up. But in host overexpression level, mice, you'll see the level stays to the same level like young animals. And of course, we look on the proteins on the cells, which is secretory cells, like goblet cells and pennant cells, which usually leads to production of the mucus and production of the proteins which stabilize the gut microbiomes. And you will see actually the number of the goblet cells gets increased in host overexpressing mice. It means one of the mechanisms how they're able to maintain this stable the gut the integrity and microbiome as well. I didn't show that data yet. Another thing, when we are looking for the more stable the gut structures of these animals, we are thinking maybe the HA is doing something with the stem cells. And we did the staining for the stem cells in CRISP and LGR5. We used the LGR5 antibodies to detect the stem cells. And we didn't see much of the difference between the control mice versus host overexpressing mice. It means it's not due to the probably number of the stem cells, but when we try to isolate the crypts and to produce the organoids, we'll see a normal increase of the organoids in aging, because during the aging, if you're isolating the crypts, isolating the organoids later, you see a decrease of the number of organoids with aging. That's what you'll see in the middle panel as well. But if your mice overexpress HAS2, then you start to see the normal level of the organoids. And the same phenomenon can be mimicked by addition of the very long form of the HA into the media and increase the number of organoids as well. And again, this is another connection of the host overexpression buildup of the HA in the tissues, and probably somehow stabilize the stem cells to keep them in more stem conditions, which allow them probably to keep more stable gut structures with aging. And this is the summary of what we observed on that host overexpression mice. Yes, we achieve relatively high level of the HA presence in the organism, and it shifted transcriptome from the older organism to a younger one, and also you have primarily reduced inflammation, which probably leads to the more stable structure of the epithelium, the gut epithelium primary, unique features of the immune response, and the protection from free radicals. But this is a nice observation, okay, but how we can go from this point into the clinic? Because we just, I show you an example, if you add HA into the mouse, then they start to see more cancer-resistant organism, probably long-lived, and we do know this is long-lived. But most interesting question, how we can take it and move it to human. Of course, we are not proposing to make it transgenic human as we did it with mice. Mice just was proof of principle for this. But I thought that in mice and human, we have a relatively high level of degradation of the HA. It means instead of boosting the expression or the production of the HA, we can try to approach the level of the degradation, to block the HA, to look for the HA's inhibitors, the enzyme which degrades the HA. And we did a screen, and we found some small molecules which leads to the high overproduction of the HA in mouse and human cells as well. And most importantly, what you see here, it's a different dose of this HA's inhibitor, the small molecules you use. It works on a micromolar range in this case, and you will see not only the amount of the HA goes up, but also length of the HA. And this is absolutely critical because I told you that the long form of the HA is a protective form which blocks the inflammation, which protects the cells from all kinds of the stress. It means right now in our hands we have the inhibitors which able in tissue culture induce the high level of the HA. What about in vivo? We took the same molecules, we injected into the mice, and now we start to see that the HA gets increased in the different tissues. And here I'm showing only two tissues because we still have the problem with the delivery of these molecules because it's not water-soluble, it's difficult to change to how to inject it. But now we have the water-soluble analogs which will be more stable, and hopefully we achieve the higher level of the accumulation of the HA in the tissues of these mice. But this is very promising. And we also use the metastatic model, the mouse metastatic model, when we injected the melanoma cells into the mouse ears, and they look for the metastasis from the ear into the lymph nodes. This is an acceptable model in the presence of the HA's inhibitor in the mice. It means the HA's inhibitor was injected like 7 days before. We are doing injection every 3 daes, and then we'll do like this melanoma models. And you can see here that we didn't see much of the difference in the primary tumors. The primary tumors will show some reduction, but it's not significant. But there was a huge effect on metastasis. Because you will see the panel on the right, you see a huge reduction in metastatic level in the lymph nodes in the same mice, which is very promising results. And again, this is the original, the inhibitor, which is not water-soluble, which is not optimal. We are going to optimize it and see if it will be much better in terms of inhibition of the metastasis and maybe primary tumors as well. And as a summary or take-home message from that comparative biology research which I showed you just right now, first of all, I want to tell you that, yes, the HAS2 from naked model extend the lifespan and the health span, most importantly, of mice. And this very long form of the HA protects mice from the inflammation and the cancer metastasis. But one of the most important conclusions for me that the comparative biology approach really works. I know because we can take the gene, we can identify the gene in different species, which protect the species from oncogenic transformation or from give it longer lifespan, and transfer it to another species and to achieve the similar results. Because I remember in the morning it was a discussion and Matt Kaberlein was talking about that we need to look for the new approaches. Here, the evolution already selected the method which is very efficient and in terms of the life extension and protection from different age-related diseases. And I give you just only one of the example of the naked mole rats. On Sunday, actually, my collaborator Vera is going to talk about whales. It means we have unlimited resources of different long-lived species, which each one of them has unique mechanisms to allow them to live longer and to stay disease-free as well. And of course, in the end, I would like just to acknowledge the people who did the experiments. It's Zhizhuang and Zhihuye, who did most of the work. The original experiments, the generation of Has2 was done by Tian Shao. He actually got a new position right now and moving forward. And we have the position available for postdocs if you want to play with many mammalian species to look for this unknown mechanism. We are working with a black box. That's what we start with, but in the end, we can find something useful for the clinical application. You can just send me emails. And I also would like to acknowledge all collaborators. Of course, all the research was done in collaboration with Vera Gorbunova, who's going to talk about the whales later in Sunday. And we have many collaborators, Stir Horowitz and Vadim Gladyshev, who is here as well. And we are working on comparative biology and different aspects of longevity. And thank you for your attention. Thanks. Yeah.
Andrei, that was lovely. Thank you so much. And I completely agree that, I mean, I'm not sure I completely agree that we are missing a lot of tricks that we could get from comparative biology, but certainly you and Vera have found a few things that have really made it work. Mm-hmm. Of course, the big takeaway that I think a lot of people will be thinking is, how come you've got this very broad benefits in terms of health of these mice, but only a really modest, in terms of effect size, extension of lifespan? So I'm wondering, have you looked at cause of death? Is there, for example, a reduction in cancer, but not other things and so on?
Yeah, that's a bit challenging because we have to look post-mortem because we have the longevity experiments and we can pick up the dead animals which was there for overnight or for some time. It means we try to do some necropsy, as you already showed. We have the reduction in the cancer, of course. We didn't do the big analysis or detailed analysis. What was the cause of death? Of course, have to be done. I think that's a good question, why we didn't see the increase. One explanation, maybe we don't see the high level of HA build up in the tissues, what we see in naked mole rat.
Well, but you would expect that to affect health span as much as lifespan, right?
Yeah, exactly. But health span benefit was much more advanced. I don't have the clear explanation why it was that there's the different effect on the lifespan versus health span. Yeah, maybe it's much easier in this case manipulate the health span and the lifespan, but this is pure speculation. We don't know. Primarily because we are thinking about the HA is one of the components of also stem cells niche. And originally we were thinking majority of this mechanism will be through the preservation of the stem cells. But we didn't see that. It means we looked on the gut, we didn't see we looked on the other tissues as well. The effect on the stem cells is low. That this will be effect on the maybe better lifespan. But you know, the reduction of the inflammation is probably that's the key differences, because inflammation is really associated with health span. You know, it's also associated with lifespan eventually, of course. But maybe because we repress the inflammation drastically, that's why we see the effect on that.
Okay, I'm going to be selfish and ask a second question. Yeah. So the data that you showed towards the end on the benefits of the HAAs, that was not in your transgenic mice, right?
No, it was in the wild-type mice.
Right. So yeah. So you're seeing clearly benefits in terms of, well, I mean, the species benefit is twofold in terms of the length of the HA and also in terms of the abundance because of the HAAs. Right. So have you considered inhibiting the low molecular weight, the endogenous mouse gene at the same time as, you know, obviously having the transgenic?
Yeah, that's a little bit challenging because the synthesis of low molecular mass, HA, is done by the Has1 enzyme. They're very similar. They inhibit one, not inhibit the other. It's challenging.
Well, I mean, you could do it genetically. You could knock it out. Or
yeah, that's probably. But we were thinking we'll better block the degradation because the two things how the short HA can be produced. One is the synthesis, and the second is degradation of high molecular mass. And from what we observed in the inhibitor work, it looks like the majority of the short form of the HA is produced through degradation. Maybe not through synthesis. It's really like preliminary data. It's my speculation and a little bit of preliminary data as well. It means, yeah, we're thinking the degradation is a more productive way to go.
Hi, great talk up down here to your right. Yeah. I was just wondering, with the effects you saw on primary tumor, which really wasn't great, versus metastasis, if you allowed the Mets to grow and did the treatment, would you see the same effect? Or I'm just thinking, could it be EMT it's blocking?
Yeah. The inhibitor which we use is a small molecule, but it's not optimized, as I already said before. We have to solubilize in DMSO, and the injections doesn't deliver everywhere, especially it has to be going into the ear. That's why we did the injection of the primary tumors. And even though we do see some reduction of the primary tumors, but it's primarily metastasis. Because I think that the trick with the HA, how it works, if you have the very aggressive tumor or like fully formed tumor, it's very difficult to stop because we are talking about the regulatory mechanism, which binds the receptors and slow down cell proliferation. If you're talking about extremely aggressive tumors, I don't think it's going to work on this stage. It means the HAs really works maybe in the earliest stages of the tumor when it's just formed and it's still not aggressive and can be eliminated. Or when you have the metastasis and the cell's trying to find the niche where to end up, that can be prevented by the presence of the HA. It means when you're talking about the fully grown tumors, it's maybe a little bit more challenging. That's possible, yeah.
Yes, thank you. Building on the discussion this morning, do you think that naked mole rats have a maximum lifespan? That is, if they were to be observed for, say, 50 years, 60 years, would we see an acceleration of mortality, or would you expect to see more of a constant mortality rate kind of along the lines of what's observed at 30 years?
Yeah, that's a good question because, it's a difficult, it's all speculation. Our oldest animal in our colony is 17 years old, which was born in our colony. We have it for 17 years. That's our oldest, and that's what we know for sure. Shelley Buffenstein, she had this oldest live, but she started from her PhD. She was like a postdoc, full professor now. She's not close to retirement, or somewhat close to retirement. That's the whole career it takes to build that collection and to see that lifespan as well. When people talked about the negligible senescence in a naked mole rat, when you have the young animal and old animal, you can clearly see this is the old, this is the young, because they are more pale, they look different, it means. You still see the senescence, and I think probably Steve is right in this case, because if you can keep them longer, they'll probably start to increase the mortality. You start to see that maybe it's the same example when you're just looking on the shorter window, just covering the earlier stage of aging. And yeah, but that's all speculation. We don't have much of the data to support that.
All right.
Thank you.